Well, welcome back everyone to the 2020 Small Farms webinar series hosted by the University of Illinois Extension's Local Food Systems and Small Farms team. This is Zach Grant again, moderating and presenting today. We appreciate you joining us for these webinars, and we'll do our best to begin and end with the space of your lunch hour. I do have a couple other educators and coordinators on who will be helping me with the Q&A, but any of the questions that you have that come up, please focus those into the Q&A box and leave the chat box for any technical difficulties that you might be having. This presentation is being recorded, and I'll email you the link to the archive presentation as soon as possible after this concludes. There'll also be a short online evaluation as usual, and you'll see the QR code at the very last slide today when we get to the question portion that you can scan with your smartphone. Again, we're using Zoom webinar, so there's just a few features for those of you who aren't familiar with it. You should see the polling question that pops up. We're collecting voluntary demographic information, and then also the Q&A box, which is a little different than normal Zoom meetings, and that's where you can enter in all of your questions that you have as we move through. I will see the Q&A box since I'm presenting today, but I'll probably, unless it's a quick question, I'll try to just save that for the very end. So today I'm actually going to be presenting. So you guys are used to getting emails from me, used to hearing my voice moderating, but my name is Zach Grant, and I'm a local food systems and small farm extension educator up here in Cook County in the greater Chicagoland area. And I primarily focus and work with urban agricultural stakeholders, primarily in sort of the focus of food production systems, as well as food safety. My background, I've been involved in small-scale farming systems for about 20 years, and in particular, I've been really interested in season extension and year-round production in high tunnels for the past 20 years, or about 15 of the last 20 years, actually. And so I'm very familiar with the different soil testing options that are out there, and was always a little dissatisfied with the traditional field soil testing methodology in our high tunnels, noticing major differences between how we're managing our high tunnels and field open air field production. So that's what kind of led me to the saturated media extract testing as a possible alternative to traditional field testing in certain contexts. So that's what the sort of theme and background for this presentation is. So we'll go ahead and jump right in. So this is a presentation that I've developed primarily working with protected culture or high tunnel based growers is what the system has been based on. We'll talk about the context of it in a second. But I'm also hypothesizing that a lot of the urban growers that I work with who grow in almost pure compost based systems that don't have a lot of soil mineralogy constituents in their media they grow in, this might be a good alternative test for traditional media tests, and we'll kind of cover that. The other thing I want to know also why I included this slide is I arranged this with some of my slides, but a lot of this is actually owed to the work of Dr. Bruce Hoskins from the University of Maine and Extension Specialist Vern Grubinger at the University of Vermont Extension, as well as a number of other Extension colleagues of mine in the northeastern part of the state. But Bruce and Vern have been big advocates for the alternative saturated media extract testing, particularly for high tunnel growers. And I'm kind of taking that with a pilot project this year and expanding on it with these compost-based growing systems. So a lot of what we're referencing today is based on their work. So you'll see their slides referenced in different parts of today's presentation. So what we're going to do just really quickly is give you an overview of what we're going to cover. So I'm going to talk a little bit about the context of saturated media extract in terms of where it's used in different production systems and the problems encountered in those production systems and why it's a good bet potentially for those systems. We'll review the SME concept as opposed to the traditional field soil test concept. Look at testing options, sampling, analysis, protocols. We'll look at some examples. And then I'm going to provide some new updates as it relates to SME, particularly in high tunnels and protected culture, because I actually have reached out to Dr. Bruce Hoskins recently, and he's provided me with some updated thinking and updated information about how they're using SME now kind of in tangent with regular field soil testing. We'll look at costs. I want to talk a little bit about tissue and petiole testing as it relates to this larger suite of potential testing tools available to growers. And then I'll briefly talk about our Cook County saturated media extract pilot project, and then finish it up with a few final thoughts. So the protected culture high compost input production systems are unique systems, okay? On the left, you'll see images related to high tunnel production where we're growing in these systems virtually year round. So we're really taxing and maximizing our output from these soil-based systems, and which typically means growers are adding especially organic growers, are adding high levels of compost, other organic matter inputs, and fertilizer inputs to make up for this, high temperature, high soil mineralization, high yield output system. So it's just a little bit different than outdoor productions. These images are from the Sustainable Student Farm project that I used to manage down on our main campus, and we would really push these tunnels to the extreme. We would get at least 3 to 4 crops per tunnel unit per year. And we paid very close attention to soil fertility in these tunnels because we realized we were really cropping down nutrients out of these systems. And at the time, I wasn't using SME or alternative soil testing techniques, just using standard field testing. And we'll talk a little bit more about SME applicability to high tunnels as we go along. Now, my current work, I still work with high tunnel growers, but now I work a lot with urban ag systems. So these are smaller scale footprint systems where growers are really maximizing their square footage output, in some soil-based systems, but also growing in other types of contexts too, as well as other techniques using tarping and low tunnel techniques that are excluding rainfall the same way that rainfall is being excluded from high tunnels. And that'll be important here in a second. We'll get to that. But the other interesting thing about urban systems in particular is that there's issues related to environmental contamination that allows growers or forces growers into using soil or media inputs that they bring from other locations. So in this case, this is our program coordinator, Sarah. We're developing what I call some compost berm bed systems. Okay, so this is a common structuralist growing media system for urban growers. This media you see here is actually a 50-50 compost and soil mix, but oftentimes growers are growing directly in pure compost and a lot of times pure animal manure-based compost as well. So growers in those systems, I'm often hearing through key informant interviews that there's lots of fertility issues and growth issues that they're having, and I think it has to do with the way they're managing fertility in those compost-based systems. And I'm actually hypothesizing through our pilot project that the SME test might be a better test indicator to help growers in urban settings growing in high compost systems manage their fertility a little bit better. The other thing to think about with these urban systems, and really any grower who is using particularly manure-based compost that have high levels of phosphorus and nitrogen and using lots of it. So runoff and eutrophication downstream of aquatic ecosystems is still a problem. Even for organic growers or urban growers who are using high levels of manure-based compost. This old idea or adage that more compost is always better might not be applicable to animal-based, manure-based compost because high levels of phosphorus loading, that the plants aren't taking up that much in season, and a lot of that negatively charged phosphate ions are moving away from that surface. In the case of this picture, you can actually see the puddling taking place, and if I zoomed in a little bit more, you'd actually start to see some algal blooms that are forming in these ponds. So even though the footprint of urban ag and these high compost systems is relatively small at the current point, if we were to expand these systems and urban agriculture becomes more widespread, the use of high phosphorus compost could become a problem the same way that phosphorus fertilizers and lawn fertilizers is becoming a problem. There's a lot of communication and education surrounding that. So an SME, saturated media extract testing, could be a good option for figuring out what your soil soluble phosphorus levels are and help you make decisions as to whether or not you should be adding more phosphorus or not. So we'll get back to that. This is just a quick slide that I use in my urban farmer training program to outline the different custom mixes that growers, particularly in urban growers and some homeowner growers, use to grow directly in. So a lot of times you will have some compost soil sand type mixes that have clay mineralogy in them, but a lot of times growers, particularly community growers and school garden growers, are growing, and a lot of commercial growers too, growing either in pure compost or some sort of peat soilless based media potty mix style mix that has combinations of peat, compost, and fertilizer. And this is also common for rooftop farming systems as well. That last growing media and pure compost, really more than likely the saturated media extract test will be a better indicator of soil fertility management decisions versus traditional soil tests. And we'll kind of get to that in a second. But I just wanted to kind of highlight the difference between, you know, these sort of created or engineered medias versus traditional soil, native agricultural soil. So again, just to highlight a few differences between protected culture and high compost input systems from open field production. Again, with the protected culture or high tunnels and greenhouses growing in soil, you're typically experiencing both higher air and soil temperatures, so that equates to faster growth, enhanced mineralization, particularly of organic nutrient sources. So growers often compensate by adding a lot of organic matter via compost or other types of organic inputs. There is no natural rainfall. Irrigation is necessary and allows for fertigation and cuts down on certain disease pressures, but there's an issue with that, mainly with salt buildup, and we'll get to that in the next slide. And in the other column, you see with high compost organic input culture, particularly for small farms, they're taking, usually this more compost is always better approach, which isn't always true, and often are amending soils with high organic matter inputs or organic fertilizers in this case. Sometimes beds are used with low tunnels, tarping, and other mulches that might exclude rainfall the way that protected culture. And some systems are based directly on growing in a compost-based media. So some of the problems that this can actually create. So in protected culture, again, we've talked about the faster growth, the quicker nutrient drawdown, but salt accumulation is always a perennial problem with high tunnel growers, particularly high tunnels that are fixed in one location. The increased evaporation pulls the nutrient salts to the surface, and if the plants can't take them up, they form this sort of white crust on the surface. And we'll get to some management implications for that a little bit later on. So things that typically don't carry over, like nitrate, which is an anion, which isn't closely bonded to the soil, which in outdoor production can move quickly. In tunnel systems, actually, you can have buildup of nitrogen and carry over from season to season. Okay, so when we'll get to some soil testing options in a little bit. So monitoring both salt in the form of electrical conductivity and nitrate in tunnels, even if you're using a traditional field test, is important and often not communicated from soil labs. So no natural rainfall to leach these salts. So monitoring irrigation is important and trying to maintain as close to soil saturation as possible. We'll get to that later. Fertilizer inputs are often unevenly distributed, and we'll get to that a little bit as well, because that's a big challenge. With the high compost input, again, this idea that more compost is always better can often lead to excess nutrients, particularly with things like phosphorus with manure-based compost, and lead to, you know, downstream effects as well as potential salt damage if the buildup is wide enough. Okay. So before we get into talking about the difference between SME and traditional soil testing, let's do a very quick soil water root system refresher. Okay, so we're not going to get too in the weeds in terms of soil science, but this is an important concept to understand the distinction between saturated media extract testing or greenhouse media testing and traditional field testing. So the concept here is to understand that most nutrients are taken up as water-soluble ions, okay? So there's a smaller proportion in a solution at any given moment. So if you look at the image on the right-hand side over here, if you follow my cursor, the blue dots represent a sulfate anion, so that could be nitrate or boron, but in this case it's sulfate, and the red dots represent calcium cation with the positively charged anions. So the only thing that the plants are actually taking up are what is in soil solution at any given time. So that typically those nutrients are moving with water through the concept of mass flow, and that increases as transportation increases. So the more water that the plant's uptaking, the more nutrients move to those root hair spots, interception takes place, and then uptake actually takes place. Diffusion, you see in the bottom left-hand side, so fertilizer granules or slow release fertilizers. Diffusion, you know, nutrients can move towards root hairs very slowly in that fashion, but most of the nutrient uptake in soil solution is moved through mass flow. So it's important to remember that concept of what's in soil solution at any given time, and that water is very important for the transport of those nutrients to the root hairs so they can be taken up. Now, they're replenished in the soil solution as needed from the cation exchange sites. So this is mostly represented by the clay mineralogy of your soil. So you see the clay particle. If we've taken a soil science class, we know how important clay particles are for those exchange sites for things like calcium, magnesium, and potassium. And as they're uptake by the plant, more is moved into soil solution. A lot of it depends on pH and clay mineralogy. Now, the organic matter in your soil, the stable fraction, like we learned last week, can have high levels of cation exchange capacity, but it's often pH dependent. So it's not as simple as, pure organic, high organic matter soils always have high cation exchange capacity. It's also a little bit pH dependent as well. And there's other, obviously the microbial and biological activity in the soil, increasing mineralization is an important part of those nutrients on clay particles and organic matter being released into soil solution through mass flow and then plants uptaking them. So it's important to remember that. And we'll kind of reference this picture right here as we move forward. So with traditional soil testing, what traditional soil testing is doing, it's measuring your capacity, the capacity of your soil to supply nutrients over an entire season, or even possibly 2 to 3 seasons. So when you get your results back, you get, pH, organic matter levels, the major cations, so potassium, magnesium, and calcium. You get some level of cation exchange capacity, and that's kind of your base soil test. And so it treats the soil like a depository of nutrients, so thinking of those clay particles as your sort of storehouse of nutrients, and it often correlates with uptake over a given season for yield, okay? The other thing about traditional field soil testing is that they're often regional tests, okay? So sometimes there might be specific regions that have certain textures or clay mineralogies that are different, and all of this actually revolves heavily around phosphorus primarily, because the modified Morgan, Malic 3, Olsen, ammonium acetate, and Bray 1, strong Bray and weak Bray testing often focus around your soil type and region and how they're analyzing phosphorus. But the important thing to remember is that when they do the test, when they take the sample, the way they handle it is a little bit differently. And then what they use for extracting the nutrients from that sample is different than saturated media. So in this case, they actually use an optimal chemical extraction. So depending on your soil test, that could vary. In some cases, it's a strong acid, like in the case of the strong and weak Bray test. In other cases, it might be ammonium acetate. Sometimes it's acetic acid, but either way, it's some sort of, acidic chemical extraction that goes in and cleaves off all the possible nutrients from all the possible exchange sites to give you a view of what is potential from your soil over an entire season. Okay, so that's traditional soil testing. Saturated media extract testing is a little bit different. This is not a new testing system. It's been around for decades, and it was traditionally utilized for greenhouse media bench crops. So greenhouse growers who were using media that didn't have a lot of soil mineralogy in it, but applied either water-soluble fertilizers or slow-release fertilizers like Osmocote. They wanted to be able to monitor their salt levels, pH, and how much fertility was in the media to make management decisions. So the saturated media extract test was developed specifically for these sort of soilless media type systems. And rather than using, so the collection process is somewhat similar. The handling process is a little bit different, and we'll get to that in a little bit. But in terms of the extraction process, the difference between SME and traditional field testing is that SME only uses deionized water. So they do not introduce typically any sort of acid or chemical extractant to pull out, the full potential of nutrients in that media. It just represents the nutrients that are in soil solution, like we just discussed a few slides before. So this high proportion of free salts or free nutrients available when you introduce that water, okay? So it's almost, the other analogy I like to use, that it's almost more similar to what the plant roots are experiencing in real time, right? There are carbonic acids and other acids that are interacting in the soil, but typically, soils are only interacting with water, right? And then the mineralization properties of the soil itself, and that's what the roots are experiencing. We're not introducing, hydrochloric acid or acetic acid into the soil to, boost the release of nutrients, right? This is more, so the SME is more a sort of short and medium term what the roots are experiencing of the nutrients in soil solution at any given time. Okay, so traditionally used with conventional bench crops, but individuals like Hoskins and Gruminger think that it could and have been advocate for using it in high tunnel systems, or systems like irrigated desert soils where there isn't a lot of clay mineralogy and the nutrient uptake is really dependent on what's in soil solution. So that's just kind of a brief overview of the difference between the two. So this is a little more in detail about the difference between the two. It's sort of the fundamental differences in those nutrient pools, right? So again, with field soil testing, it's more about what is exchangeable and available over a longer term. SME is a little bit more about water solubility. So typically with dry field soil testing, a low overall quantity will correlate well to a low intensity, but oftentimes when you have a soil test that's showing that you have high quantity or high levels of everything, which would be the case if you did a traditional field test with, let's say, a compost-based media, it'll tell you have a high quantity, but it may not reflect the intensity or water solubility or what's available in the short and medium term. So it's just a different way of thinking about the total nutrient availability in the soil. And it's reported differently. Traditional field test is usually reported on a dry soil basis. So the samples are dried and grinded and sieved before extraction. And it's measured in milligrams per kilogram or parts per million. With SME, it's an extract concentration in milligrams per liter, which is also parts per million. So here's just a quick comparison between the two. This was a study done by Bruce Hoskins in Maine, looking at both the field soil test on the top and the SME test on the bottom. So you can see, if you look at, let's say, potassium, for instance, with a traditional field test where the extractant for the sample is an acid, you can see the levels of potassium reported are very, very high. Now, according to the rating that you might get back, that's a medium level of potassium availability. With SME, with just the water extractant, you can see the potassium levels are 10 times lower than that. And according to their rating for the SME test, that's a low rating for potassium. So you can see there is some correlation in rating between the field test and SME, but the levels that are reported are significantly different. So you can't, it's like comparing apples to oranges. Both can give you information if you did both testing, but you can really only make recommendations sort of based on each individual test. But it shows you that there's a big reporting difference between the two styles of testing. So which test should you use? Okay, so we're going to spend a little time looking at different possibilities. In general, field soil testing system, they analyze it differently, so you have to interpret them and make recommendations differently, like I just discussed. For newer tunnels, a routine field analysis will work well, but I would also add on an electrical conductivity test and possibly a nitrate test, which aren't part of a traditional field test. You have to add those on typically. But as you move from year one, particularly in a fixed high tunnel system, these systems are continuously covered, and you're seeing high levels of EC, you might consider adding in the saturated media extract testing with your field soil test, or you could just do SME alone. Now for a while that was the recommendation, but now, as you'll see at the end, they're moving towards them being complementary tests. And there's ongoing research and field validation to look at different recommendations based on saturated media extract. Some of the stuff I'm going to show you today is based on high tunnel, but what isn't developed yet is really the compost-based production systems for urban growers that I hinted at, and that's what our pilot study is really going to focus on. Is SME or saturated media extract testing a good alternative or complementary test to traditional field testing? So again, for established houses, maybe a year or so old, monitoring for salt buildup. So definitely adding that electrical conductivity test, and then also adding that nitrogen test, okay? Because an open field soil and management doesn't necessarily apply because there is leaching and nitrate is highly mobile. But in a covered situation, there will be nitrogen carryover. So you can expect, the nitrogen that you added the year before, if there isn't cropping down of those nutrients, there will be carryover. So a traditional field test is fine to start, but then thinking about adding the EC and soil nitrate as a next step. So, these are based on the University of Maine soil testing packages. So they have a test, a hoop house high tunnel package test as a hybrid test, where you do the field test to address upfront potential differences. And then you start to add in EC or SME to monitor the total nutrient salt buildup. And then as a final option, the long-term high tunnel package that the University of Maine recommends for continuously covered houses, multiple years, that the saturated media extract initially might be a good test at that stage, and then add in the organic matter test, because a standard SME test does not include an organic matter test. You have to add that on, and we'll look at some costs for that in a little later. And then at that point, you're just measuring the water-soluble nutrients, because you're depending on that carryover for all nutrients. And then there is a set of interpretation guidelines we'll get to based on UVM and the University of Maine. So what does this look like in terms of soil sampling methodology and analysis? Let's kind of cover that very briefly. So in terms of soil sampling, there are a few differences, but not drastic differences. We're still taking, a number of random samples depending on the area and management area, avoiding low spots or areas that might have been over fertilized, trying to get a uniform sample from the site. So that's represented by this sort of zigzag pattern that we often reference for smaller growers in particular, where you're just kind of, randomly selecting, 8 to 12 subsamples from a certain area, collecting all that together in a bucket, homogenizing that, and that's where you get your one representative composite sample from. Now, typically, we're still doing this in the top 6 to 12 inches. 6 inches is kind of the common, but you could go down to 12 inches, but that's going to throw off your part per million conversion to pounds per acre. So if it's 6 inches, you multiply part per million by 2 to get pounds per acre. If you sample down to 12 inches, you multiply it by 4 to get pounds per acre. The thing is, it's consistency between years, consistency between management systems and different areas. So we're still collecting, random subsamples to get one large composite sample. So different ways you can take it. I mean, obviously there's, designated soil probes and augers, but you can still use a traditional spade, hand trowel, that can go deep enough to get that sort of 1/2 inch thick slice that you collect for your individual subsample. So a few other differences with saturated media extract soil sample procedure from traditional field testing, okay? For both protected culture and outdoor production with SME, testing early in the season is ideal, especially for organic fertilizer inputs if you want to know your corrections ahead of time. So if you can get a good idea of where your levels are at, then there's still maybe time to add inputs and then retest again with SME to see if you've brought it up to the levels that we're going to see here in a little bit. Early testing also prevents over application. So if you're always just basing it off of what you've always done, you may be adding more compost and more organic inputs than you need, and you're going to run into that carryover issue and having excess of nutrients potentially. So, and this is especially important with protected culture or high tunnel systems, because there is no natural rainfall to kind of leach the majority of that out. So if you're starting with a media that you also know is low in fertility, so you just know that the nutrient levels are going to be low, then you could also, incorporate amendments like peat, compost, and some of your upfront fertilizer, and then sample for saturated media extract after you do that to see if your, short and medium term water soluble nutrients are where they need to be to get you through at least the first part or most of the season. The other distinction is that the soil media should be warm and moist for a week before SME sampling. So you definitely don't want to do it when the media is too dry. It needs to warmed up a little bit and be a little moist before sampling. You don't want to let the sample sit around and dry out. That will affect the results for SME. Whereas with traditional field testing, soil temperature isn't as critical. And a lot of times people dry their samples out before they send them in for traditional field testing. With SME, you want to not dry it out. It has to be a little moist. And you want to keep some of that moisture in the sample before sending it off. So in terms of the extraction process, it's very similar to traditional field test, other than the two things we just mentioned in terms of drying and the moisture level. What happens is they'll take the subsample, put it in some sort of cup, and then it's called saturated amino extract is because the idea is that you then re-wet the sample or add more water until you get the sample to the saturation point. And again, this is a little objective depending on the media that you're growing in, whether there is some soil mineralogy in it or not, or depending on the compost or media, that saturation point is relatively objective. So you can't say, add 25 mils of water to 50 grams or a process like that. It's somewhat objective, and that is a source of error for this. But as you get better at doing this as a lab technician, that's where you want to refine your skills. Then you let it sit for an hour, and then after that hour, that's typically where, from this paste that you see here, the pH reading and electrical conductivity are measured with benchtop meters at that point. And then to get the extractant to the point where you can analyze the nutrients, then you have to attach what is called the Buckner funnel and paper, and you would add that paste to the top funnel, apply a vacuum, and then you would extract out the supernatant or filtrant that you would use to analyze. And then that extracted solution is actually analyzed. So this process is somewhat similar to field testing, but the extraction material in this case is deionized water with a traditional field tested some form of acid or chemical extractant that they use to get to this point. And then at this point, this is where the analysis comes in. Okay, so if you can send your SME test, you can send your sample to a lab, just like with traditional field tests. And just like with traditional field tests, they'll use typically what's called inductively coupled plasma mass spectrometry, or some form of ICP. And you can see this machine on the left. This is a Varian ICP-MS. This is probably a $30,000 to $100,000 piece of equipment, very expensive piece of lab equipment. That's the typical analysis of extraction. Now for pH, electrical conductivity, and nitrate, there's well-validated documentation of using things like selective ion electrodes and photometers to analyze extractants. So part of our pilot project, what we're doing with Extension is purchasing some of this less expensive selective ion electrode and photometer equipment using the photometers use what they call it's a colorimeter. It measures the color absorbance with light. Purchasing less expensive equipment and kind of trying to validate it against the more expensive ICP-MS. I'm very confident that we'll be able to get it for pH, nitrate, and electrical conductivity. For potassium, calcium, phosphorus, and magnesium, which will be the nutrients we'll use for our pilot testing, I'm a little less clear how effective these ion electrodes are. But that's going to be our process. And selective ion electrodes have been used in the greenhouse industry for a long time. And part of the reason why I'm doing the pilot is to validate how effective they are in making fertility management decisions, because these aren't cheap. This Lockwood twin meter here, the pH, EC, and nitrate meter are anywhere from $200 to $300 a piece, depending on if you buy them new or used. Greenhouse growers, again, use them. They can also be used for petiole sap analysis that we'll get to in a second. But where our project will vary from a traditional commercial lab is the analysis of extraction. Okay, so let's look at a quick soil saturated amino extract. If you sent this off, what you would get back. So this is an example from A&L Great Lake Labs in Fort Wayne. I'm going to show you a few commercial labs. I'm not, as a state employee, trying to advocate for any of these, but I'm just kind of showing you some local options, at least here in the Midwest. Depending on where you're at listening to this webinar, you may check with your lab to see if they do something called saturated media extract or greenhouse media testing if you're interested in this. So in this case, you actually see the pH, electrical conductivity, nitrate, and then all the nutrients that it analyzes. And it'll give you the result in parts per million or milligrams per liter, and then it'll give you that rating. So you see that rating on the sort of right-hand side, and it gives you kind of where they think it should be based on, if you see on the bottom there, this fact sheet that they put out, which is based on an MSU guidance from, I think it's Dr. Warnke, who developed this system at MSU. And it's actually based more on greenhouse media using conventional fertilizer. So So you're going to have to take this and interpret it differently if you're using organic inputs or slow release fertilizer inputs or compost inputs, and we'll get to that in a second. But this is the general layout of what a saturated media extract report would look like. And we'll see another example here in a second. Okay, so in terms of when you get your results back, what should the optimum ranges be? So again, the initial test developed by Warnke at MSU, this greenhouse media test, had this range that you see on the left. And then there was another book put out by Whitwer and Honma looking at greenhouse tomato and cucumber production, and they developed, you know, a separate set of optimum ranges for pH, EC, and some of the nutrients. But the current, well, this is not current anymore, but it's somewhat close, as we'll see in a second. The guidelines developed by Hoskins and Grubinger for high tunnel producers, you can see it, they've altered it a little bit. So they're allowing for a little tighter pH range, a slightly larger nitrate range, a much lower phosphorus range for SME. So this is, again, remember, soil solution available phosphorus, not total available phosphorus. Potassium and so on and so forth down the line. So again, the optimum ranges, at least in 2012, were what you see on the right hand side. I'm going to show you a more current thinking in a few slides. But again, you would just get your analysis back, compare it to these levels, and then you would want to, if you need to raise those part per million levels from your SME analysis results, here are some guidelines to help. So this was adapted from Whitmer and Honma, but this was some of the work that Grubinger and Hoskins have done, particularly with tunnel growers, and particularly for, in this case, organic growers using kind of slow, medium, and in some cases fast release nutrient inputs. So, and it's all based on pounds per square footage rather than pounds per acre. So they give you a guideline of, for nitrogen, if your levels are low and you need every 10 part per million raise that you need, this is how much fertilizer, organic fertilizer, you would have to add per 1,000 square feet to bring your levels up by 10 part per million, okay? And I have some handouts affiliated with this, and I'll actually share these both in the email and I'll put them in the link for the YouTube channel. So some of you are probably wondering where I can get access to this info. I will provide you with some of these handouts if you're interested in this. So let's go ahead and look at an example of how I actually applied this. And so I know this is a little messy, but this is kind of the way my sort of thinking with this, and you just kind of have to follow me along here. So this is a report from a grower. It's a suburban grower growing in a high tunnel. One of the problems that this particular grower is having is they feel that their pH is high, and 7.5 is kind of borderline, but the water that they use is actually Great Lakes water and typically has a higher level of calcium in it, right? So that would be a more alkaline based water. So we started with our discussion about how that pH might be lowered, and I gave some recommendations in terms of, obviously sulfur is one option, but we were talking about the possibility of peat as an organic matter input with no fertility tag-alongs. And then also there were some questions about acid injection into their irrigation system. So I'm not going to diverge too much into that, but if you're interested in that, I can provide you some information about direct acid injection into your irrigation water to potentially lower your pH over time in your soil. So in this case, with this report, I just went down, took the rubric, or at least this is actually the newer rubric that you see here where it says Northeast growing recommendations for high tunnels. I drew those in, and then I compared it to the result. And you'll see that in a lot of cases, the result from the high tunnel recommendations differ from the A&L lab's recommendation. And that's because they're based off of two different systems. They're close in some cases, but in some cases they're different, particularly with potassium, as you'll see. They're saying that it's acceptable, but according to what we saw on the previous slide, this slide, the potassium should be between 150 to 275 part per million. And in this case, it's 94. So it's in my eyes, it's actually a little low. So when you look down everything here, everything is okay except for nitrogen and potassium. So in this case, the only recommendations I would be making to this grower is that you're obviously going to have to input some nitrogen, which we're all probably going to have to do anyway. But you definitely need to bring those potassium levels up, particularly if you're growing any fruiting crops in here, because those tend to be high potassium consumers. So in this case, I was making the recommendation of blood meal, okay? Because I didn't want any P or K tag-alongs, and I was going to use potassium sulfate or potassium sulfate for the potassium recommendation. So what I did was I realized that the difference between, you know, the high level of nitrate and what we have, 24 parts per million, so 200, right, is the high level recommendation. We have 24. That's a difference of 176 part per million. So what I did was I went in, divided that 176 by 10 part per million, because that's how many of these units we're going to need to bring it up, 17.6, and then I multiplied that by 4.2, because that's how much you need to bring it up by 10 part per million. And that's where I came up with that approximately 74 pounds per 1,000 square feet is what I would need to add to this growing media to bring the nitrogen. It was what I would expect if I applied that, then repeated the SME levels, they should be much higher. Okay, so it's kind of a range. It's not exact, and most fertilizer recommendations, even from field tests, aren't exact. They're trying to get you into a range rather than giving you an exact quantity. And then I applied that same logic to the potassium recommendation that you see over on the right-hand side. So we have approximately 94 part per million. I want it to be 175, so I need to bring it up by 81 part per million. And I applied that same logic with potassium sulfate, okay? And that's where I came up with 4.5 pounds. So that's what I would make my recommendation here, is I wouldn't be adding any more compost, because that might be tagging along unneeded nutrients like phosphorus. You can see the phosphorus levels here is 9.8, which is, according to this rubric for high tunnels and SME, is very high. It's double of what it should be. So you definitely wouldn't want to be tagging along any phosphorus in the form of manure-based compost or any other fertilizer. So that's why I was very specific about blood meal and potash of sulfate in this case. So this is kind of the logic that I would use in analyzing SME and giving this information to the grower. And part of the service that we would use with our pilot project is to do just this. So whether we're using our testing system or verifying it with a lab, we would make these recommendations and provide that technical assistance to those growers. So the newest recommendations for this system are as follows. I just talked to Dr. Hoskins last week, and he provided me some newer information. And what they're doing now is, rather than just relying on SME for high tunnels, they're actually recommending definitely starting with a standard soil test, especially in the beginning, to see if there's any big imbalances that you need to correct. And then also using SME to kind of correct some real-time nutrient availability, okay? So using kind of both tests to verify. And with the standard soil test, you probably, and I haven't verified this yet, but my thinking would be is that you would do that probably once a year, maybe every other year. But the SME test could actually be done maybe more than once a year to kind of make sure that that short and medium term soil solution nutrients are where they need to be for optimal plant performance. Now, these charts on the right were developed by Vern Grubinger, and they're similar to the metrics that we just went over in the last example, and I've tried to compare them. They're close enough in terms of the recommendations, but if you kind of read through some of this, you know, like for potassium, for instance, they're recommending, here's the application rate based on the modified Morgan field soil test. And then on the bottom, it says, if your concurrent SME test shows, you know, a certain level of phosphorus, if it's less than 1 part per million, then you should also add that as a starter fertilizer. So they're kind of, you know, tweaking their recommendations a little bit now based on both a standard test and your SME results combined. And I think that makes sense to kind of give you a whole picture and help you make better fertility management decisions. So I can, again, provide you with links to these tables if you're interested. We don't have time to dive in the details, but there are some newer recommendations and things are kind of evolving with this testing system. But there still is relevance for saturated media extract testing, particularly in high tunnels or protected culture. So let's look at some costs. And this is the big question that a lot of people have, is what's the difference in cost? And in some cases, depending on the lab that you go to, it can vary. This is a spreadsheet that I'm working on right now. I'll probably make some additions to it and take some things off. And it doesn't include our pilot project testing, and I'll get to that in a little bit. So I'm looking at, you know, 2 Midwest labs, A&L and Midwest out of Nebraska, and then 3 land grant soil testing services. So as a lot of you probably know, in the Midwest, there's very few land grants that still offer soil testing. In the Northeast, and well, I guess with the exception of the University of Minnesota, the land grant extension services still actually do some in-house soil testing. And with what we're doing with our pilot project is trying to bring that back a little bit, at least for the University of Illinois. So you can just kind of go through this and see some of the differences. And the cost for the saturated media extract test actually vary quite a bit depending on the lab that you go to. Again, not making recommendations here in terms of what lab you should use, but I'm just showing you a few different labs and kind of the cost differences between. So you'll notice the A&L lab test, if you were to do just an SME and then you added on an organic matter test, loss on ignition test, that would bring your total up to about $48. Their traditional soil test with recommendations is around $33 with all the bells and whistles if you add in, I think, nitrate and electrical conductivity. So you can see it's a little bit more expensive to do the SME from A&L. Midwest labs that I've used them in the past in personal work, their traditional soil test with also bicarb for P and nitrate and electrical conductivity is $27.50. So pretty similar to A&L. The weird thing is they used to have what I thought they call the greenhouse media extract test. They now have something called water-soluble extract, but you have to use it as an add-on to a traditional field test. So it's only $5, but you need to add it on to a traditional test. So based on what Grubinger and Hoskins are currently recommending of doing both and comparing them, that's actually not a bad deal. So if you did $27.50 and then added that $5, you'd have somewhat of a picture of water-soluble extract or available nutrients and your traditional field test. And then as you move down, these are the extension service ones. The Minnesota lab would be more relevant for our traditional field test. The main lab and Massachusetts lab, you'll see for their traditional field test, they use the modified Morgan extraction. So I'm not 100% sure if that extraction method is appropriate for Midwest soils. You'd want to verify that before spending the money on that traditional field test. The University of Maine lab, that's where Bruce Hoskins is, and he runs that lab. He offers a really good deal where you can get the cost of the traditional test and the SME test combined for $30. So it's a pretty good discount, but again, I don't know if the traditional field test, what they do, is appropriate for the Midwest. You'd have to run that by them. And then of course you'd add on your priority shipping depending on how much sample you use, anywhere from $8 to $15, and that kind of gives you an idea of how much an individual sample will cost. Okay, so somewhere in the ballpark of traditional testing, but if you're doing multiple of these tests a year, it could add up. So part of the reason why I want to do this pilot project, particularly for our underserved Cook County urban growers, is to kind of maybe try to ameliorate some of the costs for SME, but still get some of the good information out of it. Okay, so we'll come back to that in a second, but I want to just hit on very briefly both plant tissue testing as well as petiole sap testing. So particularly for high tunnel growers and fruiting crop growers, okay, so tomatoes, solanaceous family growers, plant tissue and petiole sap testing will give you an idea of what the plants are actually uptaking and incorporating into their tissues. So the analysis is completely different. For pure plant tissue sampling, a lab will have to do it, okay? So you're typically collecting multiple leaves of recently mature, fully expanded leaves, below the first flowering cluster, put it in a special bag, sending off the lab, and they analyze it for $20 to $30 per sample. And they typically give you tissue sample nutrient ranges to affiliate with. So these ranges are different than SME, they're different than traditional field testing. This is more the optimal ranges for the tissue itself. And this, if you're meeting these ranges, both before and during fruiting with macro and micronutrients, then your soil fertility plan, in theory, is working. But if something is off here, then that's where you could, in real time in season, potentially correct or feed the plant directly via your fertigation system or broadcasting below the canopy. The petiole sap test is a little bit different, only in that rather than taking the full leaf, you're actually just isolating the petiole from the leaf, getting multiple samples, crushing it, collecting that sap. The labs will do it, but the selective ion electrode meters will actually measure petiole sap as well. So another reason why maybe purchasing at least the nitrate meter might make sense for greenhouse growers. Again, the cost is somewhat steep, but we're going to be trialing this out as well. So I've referenced this already. We're running a SME pilot using the selective ion electrodes and some cheaper photometers. So we'll do the identical SME extraction process to the lab, but we'll just analyze it a little bit different. And I'm going to be doing some verification with various labs to see if some of the potential interferences with the electrode method correlate well with the ICP-MS method. I've seen some papers that say that it does, but I've also been told that there's interferences with selective ion electrodes and photometers where you might not be as accurate as ICP-MS. But again, if it's close or if it's in the ballpark, you know, and we're looking at ranges of recommendations, then this might be a cheaper alternative. And we're primarily focusing this project this year in Cook County in my unit with urban growers. But if it's successful and we can keep the cost down, we'll probably offer this as a relatively low cost SME testing service from the University of Illinois Extension. Definitely for pH, EC, and nitrate, we'll see about some of the other nutrients. So keep on the lookout for that. So just a couple final things before we answer some questions, if there are any. I just want to highlight a couple things about irrigation and fertility and some larger thoughts in general. So particularly with potassium and nitrogen, a lot of people ask the question about front loading rather than split applications. So with both nitrogen and potassium, if you have relatively fine textured soils, right, or high organic soils, clay soils, high organic matter level soils, and you're using fertilizers that are relatively slow or medium release, front loading nitrogen and potassium at the beginning of the year is fine, okay? You can do it, especially in a high tunnel where leaching isn't gonna be as big of a problem. But if you have coarser soils, sandy soils, lighter soils, you should think about splitting those applications, either a third or a half of it up front, and then the other portion of it midway through the season. Keep a special eye out for potassium because particularly for fruiting crops, potassium tends to be luxury consumed by tomatoes. Even if they don't need it, they end up taking and cropping up a lot of potassium. So it's important to pay attention to that from year to year. The other thing about slow release or organic fertilizer inputs that is often not well understood or practice is that those materials need to be thoroughly both wetted and distributed through the bed profile. So you see some examples of people just throwing a band of, you know, like dry, coarse meal-based fertilizer. But if you want the fully expanded root system to interact with that, it needs to be incorporated throughout the entire media and it needs to be thoroughly wetted. So in terms of the wetting profile with that, you know, it really, with fruiting crops, drip irrigation is the important component. So rather than just having maybe one drip emitter line per bed, right, you would maybe have 2 all the way up to 4 drip lines per bed to make sure that you have even moisture on those bed tops. So those minerals, the nutrients are actually mineralized from those organic amendments and the plants are actually taking those up. The last thing I'll reference here is to and as you know, the continuation of that thought is that you need to keep soil moisture close to field capacity. And typically we're doing this with tensiometers, and we're able to do this with drip irrigation and timing and tunnels very well. But if you allow those fluctuations to occur, you can have all sorts of problems. Like you hear about, you know, calcium deficiency and then blossom end rot problems. But also the salt levels, if your salt levels are high and you're not maintaining good soil moisture, then that's where you're going to see, you know, root dry out, desiccation, and potentially plant damage. So it's important, especially if you have high levels of EC or nutrient salts, to keep that soil evenly moist. Okay, so some closing thoughts before we open it up for questions. We still have a few minutes left. It's important to remember, I know this has been very focused on the soil sort of nutrient or chemistry of the soil. We still have to manage the other soil factors as well, okay? So Grant talked about that last week a little bit with organic matter management and inputs. So we're still thinking about both the physical and biological properties of the soil as well. It's not just about the chemistry. We need to keep that in mind. Develop a good IPM and pest monitoring system, okay? Especially for tunnel systems. It's important. We often get misdirected in thinking that the higher yields, higher growth, that we're not going to have pest problems in these covered systems. We do, and they need to be monitored and managed. And also that will help distinguish, you know, the biotic from abiotic symptoms, because a lot of times nutrient deficiencies and problems with, you know, salt buildup, for instance, might be misconstrued as a disease problem. So having a good pest monitoring system and understanding the difference is also really, really important. Remember, the standard soil test is measuring a reserve of nutrients in the soil. The SME or media extract tests are for available nutrients, and tissue and petiole testing are for what the plants actually took up. So 3 different testing systems you can use, all important and all will help you manage your systems. Look out for high salt, high pH, high phosphorus levels in soil, media, compost, irrigation water, manures, and fertilizers. It's important to pay attention to what we're putting into our soil, regardless of if it's organic or not, or if it's compost or conventional. We need to make sure we understand what's in these inputs, read the labels, understand salt indexes, don't over fertilize. If we need organic matter, we could consider non-nutrient based sources like peat moss or leaves or other materials we can add. If we don't need to add something like phosphorus, use targeted nutrient fertilizers. Don't use fertilizers that have tag-along nutrients if we don't need them. Another thing to remember is if you do have salt levels that are building in the top few inches, which is where we typically see these high salt levels, particularly in tunnels, if you mix those soil levels a little bit, mix the top 3 inches, you can incorporate some of that salt down further in the soil profile, and that will help distill it or prevent salt damage to the roots themselves. So if you see some of that salt crusting, irrigation can help. So people ask the question, can I just apply 4 to 6 inches of water to my beds and will that leach the nutrient salts out and lower my EC? Yes, it probably will, but adding 4 to 6 inches of rain at once to a tunnel system is going to create some other problems. So we just need to monitor our fertilizer input levels, maintain good soil moisture, and we should be able to keep our salt levels under control. The last thing I'll leave you with is that soil and tissue testing or media testing is a critical factor for managing soil fertility. So otherwise you're just guessing, and that can lead to all sorts of problems that we illustrated in terms of salt buildup, leaching, especially in tunnels. And the only way to really track whether your fertility management decisions are making a difference, other than maybe some of the tissue testing I referenced, is to track your crop performance. So I know some of us are doing this, some of us might not, but tracking our yields per bed unit, per tunnel, simple metrics to see if there are differences from year to year or season to season with our soil fertility management decision. That's the only way we're really going to be able to assess if those things are actually working. Okay, so with that, we're going to open it up for questions. We still have about 4 to 5 minutes. Here's my contact information if you want to reach out to me. And here's the QR code right here that you can scan with your phone to do the quick survey evaluation feedback. So we'll go ahead and open it up to questions. It looks like we do have a couple questions. Let's see here. I'm seeing a couple in the chat and I'm seeing a couple in the Q&A. So I'll look at the chat first. Someone's asking, what percentage of high tunnels have problems with salt buildup in under and over 4 years? I don't have an exact number to give you, but I would definitely say that when you get to that 2 to 3 years of continuous cover under a high tunnel, salt buildup will start to be a problem, especially if you're adding lots of compost or manure or fertilizer type inputs. You're going to start to see it at that probably 2 to 3 year mark. So I can't give you an exact percentage, but you just need to pay very, very close attention. Someone also is asking about filtering rainwater to help reduce the Great Lakes water effect. Would municipal water treatment be affecting it? So that's a great question. I don't think I have a solid answer for you on that, but in terms of reducing the alkalinity or pH of the water, I think that acid injection might be a starting point. And when I say acid injection, it sounds scary and complicated, but all it would be is if you have a fertilizer fertigation system hooked up to your drip line or sprinkler system, you would just be injecting acid at that point. Okay, so you might, if you're also need an injector for fertilizer, you might consider a downstream injector for acid and then an upstream injector for fertilization. So you're correcting the water alkalinity and pH first and then injecting the fertilizer after, or it could be the opposite. I'm not 100% sure. I can get you some information about that. That would probably be the best way. Sulfur and peat additions might bring that soil pH down if you have alkaline water or alkaline soils or high pH soils. But again, you'd have to track with testing and management to see if it's actually making a difference. So really great questions though. So someone is asking, wondering about the percentage of salt buildup. Oh, that was the question that just got asked. So I think I kind of answered that. So is saturated media testing affected by soil bulk density? That's a good question, and I don't think I have the appropriate answer for you. only to say that I don't think it is because you're not drying the sample. So whereas part of the reason why traditional field testing is dried out is to a factor for that soil bulk density issue. Where in this case, because you're not drying it out and you're collecting a representative sort of real-time sample of the media, the bulk density really isn't factored into play. And again, you know, it's comparing SME to traditional field testing is comparing apples to oranges. So we can't really look at the two, but I don't think soil bulk density is a factor with SME. People often have asked me about the cation exchange capacity with analysis with an SME test. And, you know, again, as you saw with a basic SME report, you didn't see any information about cation exchange capacity or exchange sites. I think you could calculate it, but when we're thinking about the exchange sites and reserve nutrient capacity versus what's available in real time, the cation exchange capacity, I think, is less relevant in that case. But those are both good questions. Someone asked the question, why did you suggest blood meal versus ammonium? Oh, versus ammonium sulfate. A great question. So in this case, these are organic recommendation. So right, so if this was a conventional recommendation, ammonium sulfate would be fine. You would just have to factor in for that higher percentage of nitrate available in ammonium sulfate, and you would be adding less than the recommendation I made. So yeah, so sorry I didn't clarify that. The results or that rubric that I was using was based more on organic input fertilizer and not conventional. So with conventional, you definitely would want to cut those in half, or look at the higher percentage available in a conventional fertilizer and reduce accordingly. So good question. And then the final question I see on here is Brix testing a good substitute for more expensive petiole uptake testing? Great question. I don't have a good solid answer for that. I've heard and often discussed the concept of testing for Brix. So for those of you who don't know what that is, that's essentially, the available sugar content in a sap solution. So it's often used in vegetable production to measure maturity indices for things like melons or tomatoes to figure out if the sugar content is high enough and we've reached a good harvest point and we're ready to harvest. Now, there is some discussion that the higher sugar synthesis means that correlates to higher nutrient uptake and positive plant growth. I've never seen any data or papers that discuss this in great detail. It seems like it's somewhat anecdotal versus data-based, but again, I could be proven wrong if I were to see the data. But I don't know if it's a one-to-one replacement, but it might, if you have a Brix testing refractometer, then it might be interesting to compare the results to see the difference between the two. But I don't think it's a good one-to-one substitute for the petiole uptake testing. Okay, so those are some really great questions. I hope you guys, if there's any more questions, we're right about the 1 o'clock hour, I can go ahead and answer them really quickly. But if not, I really want to thank everyone for joining us again. And I know this topic, it seemed a little strange in advertisement. I should have explained it a little bit more, but I think it holds promise, particularly for tunnel growers, protected growers, and maybe for urban high compost intense growers. So I hope you got a little bit out of this. Maybe you can make some changes based on it. If you have any questions, feel free to reach out. Definitely, we'll get you the link to this as soon as I can. And we'd really appreciate any feedback through the QR code evaluation or anything else that you can provide us. And one last question. What soil temperature do you need to test this in spring? Another really great question. When I look at Grubinger and Hoskins' recommendation, they don't give an exact temperature recommendation. Although in a tunnel, you would expect that this time of year on sunny days, your soil temperature is starting to warm up significantly. It would definitely need to be, probably above 40 to 41 degrees would be my guess if I had to give you a number. But the warmer the better. So, if you want a sample now to help you make decisions so you can incorporate fertilizer, then go ahead and do it. But you definitely don't want it close to freezing, or frozen soils wouldn't work in this case. It definitely needs to be somewhat warm and moist when sending. But a great question, but I don't have an exact number for you. But I will write that down and try to get to the bottom of that. All right, so if that's all, thank you guys so much. And this is the 5th webinar. We have 3 more, and we hope to see you in the next couple of weeks to finish the series off. So thanks a lot, everyone.